Thank you for the introduction. Um, as you may know, in a normal human genome, there are 23 pairs of chromosomes called the karyotype. However, in a cancer genome, chromosomes are often unstable, showing a high rate of segregation errors during cell division, known as chromosomal instability, SIN for short. SIN can lead to numerical and structural chromosome alterations, such as chromosome gain, chromosome loss, and translocation from one chromosome to another. SIN plays an important role in cancer evolution. They can cause different types of structural variants, including copy number alterations. These alterations cause heterogeneity, upon which natural selection acts to enable genomic plasticity. They can also trigger cellular stresses and lead to inflammatory signaling. As a result, SIN facilitates several processes, including metastasis, immune evasion, and therapeutic resistance. Diverse patterns of SVs have been detected in cancer genomes. However, these SVs typically appear as joining of breakpoints. They are often caused by DNA damage and errors in repair. Non-homologous joining is a major repair pathway of double-strand breaks. Many SVs in cancer genomes are caused by NHEJ. However, we still don't know how the DNA repair mechanisms directly contribute to the formation of complex SVs in cancer genomes. This question cannot be answered by experimental approaches alone due to technical limitations. Quantitative models can help by encoding our knowledge of the underlying biological processes. The current models mostly focus on specific SV types and local genomic regions, so they may miss underlying processes that relate to different types of SVs. Here in our work, we developed a simple yet unifying framework to model the dynamics of SV formation based on double-strand breaks and its repair via NHEJ. Next, I will briefly show our model. We simulate the formation of SVs via double-strand break and repair in a cell cycle. We repair double-strand breaks based on their genomic distance. We allow a few unrepaired double-strand breaks, simulating cell cycle checkpoint errors. We embed this process in a stochastic branching process. We simulate the segregation of chromosomes depending on the number of centromeres. If there are multiple centromeres in a chromosome after the chromosome fusion, we can also simulate the fragmentation and the breakage of the chromosome. This is a demo showing the our model starting from a normal cell and in 2 cell cycles when 5 double-strand breaks were introduced in each cell cycle and randomly joined. Our model can help to explain the formation of complex SVs. For example, in a recent study, it shows a complex SVs such as chromosome can form after a single cell division error from this chromatin bridge. In our model, we can simulate this process. Can simulate this process of chromatin bridge formation and breakage and local fragmentation. Here is a result. It shows the cell lineage of 100 cells. The heat map shows the copy numbers of each cell. Here, the red arrow indicates a cell with chromosome with this typical oscillating copy number patterns. One important utility of a model is to fit real data. We fit real data and estimate the parameters of a model using an approach called approximate Bayesian computation. So basically, we sample parameters from known prior distributions and simulate data, compare simulated data with real data, and got the positive distribution of the parameters. We validated our model on similar data here. You can see the positive the distribution of the inferred parameters are quite close to the real values. Importantly, we can get the positive predicted distributions of two unobserved parameters, such as number of chromosome fusions and the number of extra chromosome DNAs or ecDNAs. We applied our approach to about 20 single-cell data sets from a recent paper. These are the posterior distributions of the model parameters. And the last two rows show the posterior distributions of the inferred number of chromosome fusions and the ecDNAs, which are two important parameters in real data. Our model can also reveal the novel correlation between these different parameters or measures. Interestingly, we found this CCNE1 amplification is related to multiple SVs, such as chromosome synthesis, ecDNAs, and whole genome doubling. In summary, we propose a first model of general SV formation at the whole genome level based on the fundamental DNA repair processes. Our model can recover K parameters in SV generation and reveal novel correlations among SVs. For the time, due to time limit, I didn't give much detail. So if you're interested, you can read our paper recently published. Finally, I would like to thank my previous supervisor and my collaborators, and I'm starting my own group, so I'm looking for PhDs and postdocs. If you're interested, please feel free to talk to me. Thank you.
Do you have any quick questions for Bingxin? Yeah, Roland, we have one question for you.
Question about the biology. So, when does all this genomic instability, how does this genome avoid becoming horribly knotted? Like, if you're replicating and there's bits and pieces breaking off, like, how does it stop getting knotted? Like, tied in knots, like two replicating chromosomes?
You mean how the this chromosome instability initiates? Sorry, I didn't get it.
So in normal chromosomes, I guess they're not knotted, right? Like, you know, like you get tied a knot with string. But if there's a lot of instability in the genome, how does the cell stop becoming knotted? Like that, because if it's knotted, then how does the cell divide if the two genomes or chromosomes are knotted?
Noted. Okay. I didn't quite get. Maybe we can. Okay. Yeah. Thank you.
Okay, thank you very much. I would like to thank all the speakers of the morning, and then please enjoy the lunch.